Welcome to the second presentation on muscles. So this is the number 2. We're going to talk about the excitation-contraction coupling, and then a few aspects of muscle function and fatigue, and this will cover the slides from number 40 to the end of that set of slides. So with the excitation-contraction coupling, we start with the events at the neuromuscular junction, and then end with the contraction of the muscle. Just checking, okay. So we start with number 1 and number 2 here at the neuromuscular junction. So the somatic motor neuron releases the acetylcholine at the neuromuscular junction. So this depends upon action potential frequency in the interpulse interval, as you recall, and how many pulses are sent collectively to determine the concentration of acetylcholine in the synapse. It binds to the receptors. And then, of course, these are ligand-dependent sodium ion channels, so there'll be an influx of sodium. Remember that with any action potential, just as we have here in the sarcolemma, the sodium-potassium ATPase pump is responsible for reestablishing the sodium and potassium concentrations, and the action potential generated in the sarcolemma is otherwise the same as that in a neuron. We'll see that the cardiac myocyte action potential differs, but the action potential here, this is why we're not going to discuss it again, is exactly the same as in a neuron, except now it is passing along the sarcolemma rather than the axolemma. So this is an excitatory postsynaptic potential that we are seeing here. And it triggers an action potential, so the trigger zone is at the periphery of the neuromuscular junction, and the action potential spreads in all directions along the sarcolemma from the trigger zone, which is surrounds the neuromuscular junction. So there'd be a trigger zone surrounding this neuromuscular synapse here as well, and the action potentials will go in all directions if the membrane is ready to be activated. Remember, the absolute and relative refractory periods apply to any patch of membrane here. So this portion in red that is presently depolarized cannot experience another action potential. It is in the refractory period. So keep that in mind. It's not just this little localized event, but this does show us that the T-tubule membrane is exactly the same, and so the action potential will pass through its length, and we saw how it penetrates all the way through the muscle surrounding each of the myofibrils at a triad, and this is the triad. If we put another sarcoplasmic reticulum on the other side, this would be the triad, a terminal cisternae where the calcium is sequestered, a T-tubule, and then another terminal cisterna over here. So what we are following then is the action potential down into the T-tubule, and we'll go through each of these steps one at a time. So the action potential in the T-tubule alters the conformation of the DHP receptor. This is a dihydropyridine L-type calcium channel. And then there's also a ryanodine receptor channel that we'll see here as well. So some of the labels are staying stationary and don't make any sense until we get to that point. So what we see is that the action potential acts on voltage-dependent, voltage-activated molecules, proteins, that undergo a conformational change. And in this case, we see it's working kind of like the mechanism inside most of the classic toilets, where a little cap is pulled off by this string. So this would be the toilet, the flush handle that you'd see here, and when it gets changed by the action potential, pulls and then opens this channel. So it's a mechanically gated channel in that sense, the DHP receptor. So the DHP receptor opens the ryanodine calcium release channels in the sarcoplasmic reticulum, and the calcium enters the cytoplasm. So the yellow structure here with the X and the blue molecules beside it, that's the DHP, and it has this connection, as we had said, to the ryanodine receptor channel shown here. And it opens the channel. And then the concentration gradient is tremendous. You've seen the previous one, they didn't show any calcium in the sarcoplasm period, and under resting conditions that should be the case. What they are not showing is it's scattered all along the length here within the sarcomere, within the sarcoplasm. On the surface of the sarcoplasmic reticulum is the ATP-dependent calcium pump that's going to be pulling, so if that calcium bumps into it, it goes right back in rather than being able to diffuse anywhere. But as it diffuses into the myofibrils nearby, within a toward the myofilaments within a myofibril nearby, then it is more likely to bump into and hit the binding site, calcium binding site on the troponin molecule, and of course this will cause the event that we had talked about. The myosin heads will then execute the power stroke if ATP is available for them to be all activated and ready in the state of highest affinity to bind to the myosin binding site on the actin, as you recall. You can have all the affinity you want. If the calcium isn't there to expose your binding site, you can't expose to it. As soon as the calcium's there and exposes the binding site, you bind to it, and then you complete the entire process that we had talked about, and the actin filament slides toward the center of a sarcomere. So these distances actually are known, the distance that each of these can contribute, and the calculations have been made. That's beyond our scope at this point, so we're not going to be concerned about it. But with a certain amount of calcium available, with all the ATP that would be needed held constant, you can generate a certain degree of contraction simply by putting calcium into the cell, and the same thing with stimulating it. So then we can begin to think about the change in a muscle fiber that we would measure. And remember, here we're just thinking about measuring the tension in the lower panel here in a single myofiber, not necessarily in an entire muscle. Remember, it's going to be different for the muscle because you've got the parallel connective tissue contributing as far as tension would be concerned. So here we're talking about a single muscle cell. So a twitch is a single contraction relaxation cycle. So you can have a twitch within a myofiber and a twitch in an entire muscle. So there are a few differences. The interactive physiology covers the muscle twitch in a full skeletal muscle very nicely. Here we're just considering it as a single cell that we could record the electrical potential, and here we're recording the membrane potential for the neuron and the muscle fiber. So we are following the same time scale on the x-axis for all three of these graphs, and then the y-axis differs. We have although it is the same scale for the upper panel, but that's for the neuron membrane potential, and the muscle membrane potential is shown here. So minus 70 is the resting potential for each of these, but you see that the peak of the action potential is offset by the time period required for the action potential to reach our recording electrode in the muscle cell itself. So this is to reinforce the idea that we need the action potential in the neuron, the alpha motor neuron, first in its terminal to cause the release before we can measure an action potential anywhere in the muscle fiber. This exact time interval between this point and this point only depends upon the distance between the neuromuscular junction and your recording electrode in the muscle fiber. So the farther away it is, then the greater the time between these two peaks, as you might know. But because it's an action potential, progressing through here, you won't get a decrement in the amplitude of the peak. It's going to be an action potential that you measure, just as if it was an axon. So this is not a graded potential. It's an action potential that is passing along the sarcolemma and into the T tubules as well. So it's an all or none event. So it's still a 2 millisecond event. And we can see that the action potential on the membrane of the myofiber is indicated here on our time scale. The first evidence of the positive deflection, the depolarization, is shown here, and that corresponds to the peak of the action potential in the axon terminal. So we know that something is beginning to happen molecularly at this point, and we can see that there is a period of time called the latent period, over which we do not see any deflection in the tension within the muscle. Once the tension does begin in the muscle, the skeletal muscle fiber, single muscle cell we're talking about here, we would call that the contraction phase. The timing of the latent period you can see here for a single myofiber is the same time interval of the action potential. So you could fire another action potential right here to obviously to summate on the contraction. So that's what's really important about the latent period for muscle tension, the contraction event as it is coupled to the excitation, is that as soon as you begin to the contraction phase of one excitation contraction coupled system, you can fire another action potential and contribute to the summation. So by the time we would get to here, or the latent period of time into this contraction phase is going to be the latent period of the next event here, and we can begin to build on that. So we introduce that idea at this time. Otherwise, if you don't get another excitation contraction event in this time period, the calcium is going to be removed, right? And you're going to get the relaxation phase. So this 10 to 100 milliseconds differs, and it is calcium concentration dependent. So in other words, the contraction relaxation phase represents the molecular and biophysical dynamics of dumping a certain amount of calcium into the sarcoplasm, and the rate of its distribution and binding, and contributing to the active form, if you will, of the thin filaments where the myosin binding sites are revealed. Then you get this contraction. And the reversal of that distribution of that amount of calcium by the ATP-dependent calcium pumps represents the relaxation phase. So this whole time interval represents that dynamics of putting a certain amount of calcium in, to which you will bring about a certain amount of contraction and thereby tension that can be measured in the muscle, and its reversal. So remember, assuming the ATP is present, all you need to do is to change the amount of calcium. Keep in mind that this is the single cell event with a single axonal input to it. When we talk about motor units, there's different kinds of motor units of how many alpha motor neurons are going to innervate a single muscle fiber and be recruited to fire at a certain period of time, and how many muscle fibers are within a muscle cell, and whether you're stimulating the entire nerve to that or coming from the spinal cord spontaneously. that whole idea is covered very nicely in the interactive physiology, and I urge you to watch it, and we will discuss it in general. But the important thing to keep in mind when you're studying muscle physiology is, are they talking about a single muscle fiber stimulating a single axon, or stimulating multiple axons, or are they talking about a whole skeletal muscle intact or in an experimental prep, and is there a load on it to start with? Or are you talking about a natural reflex-generated recruitment of the motor units to an entire muscle and measuring the tension generated in the muscle as a result of say, a cross-extensor reflex type of a natural recruitment of the motor units? So don't let this be so simplified of a reduction of how things work that it confuses you when we get to talking about a whole muscle, and I'll obviously bring it to your attention then too. So that makes the point that if you squirt a certain amount of calcium into a skeletal muscle cell, let's say you didn't bother with the axon, you just had this filled with some calcium, and you squirted a certain amount of calcium into there, so you know what concentration of calcium it would produce inside the muscle cell at its peak, then it would generate a very characteristic tension profile, temporal, from the time point that you squirted it in to when it would bring about the event. It wouldn't look just like this because you're squirting in the calcium rather than having an action potential bring it about, so the latent period would be artificial, but anyway, you would be able to get the contraction relaxation phase and be able to change all those dynamics, the rate of increase, the peak height, the total amount of tension you could get, and the rate of the relaxation phase would depend dynamically upon the amount of calcium that you injected. So in other words, physiologically, the higher the frequency, the more you're going to get in a single muscle fiber, right? Because the number of, to come all the way back, I know I hate to do this to you, it's all back here, physiologically. So if you send a 20 hertz with a short interpulse interval, you're going to be blasting acetylcholine here and generating action potentials really fast in a short period of time. And they all, within the limits of the refractory periods, they all can get here and bring about this event and keep opening this on a higher frequency so that more calcium can continue to come out and overwhelm the rate at which it can be returned back into the sarcoplasmic reticulum. So it's this whole dynamic of, again, this is the faucet into your bathtub, and how frequently are you opening it. Might be a hand pump that you'd have rather, the old hand pumps. So how much can you get out with every pump every time you open this, versus how quickly it drains back out of the bathtub, and therefore how much calcium is going to be available to diffuse out here and randomly collide into these structures, and at what frequency would it be bound to the troponin and be exposing the myosin binding site on the actin. So that makes it seem more complex, but that's the way you should ultimately appreciate it in your mind. So that's the calcium variable. There's two dependent variables once we get down to it. There's calcium being present, and the other one was the ATP. So how much ATP is available within a muscle cell? I was kind of surprised. The textbook says the amount of ATP in a muscle fiber at any one time is sufficient for only about 8 twitches. So that, remember, they said fiber, not a skeletal muscle. So we're back to thinking of a single skeletal muscle cell. So it can't be recruited too many times for a single behavioral event, without depleting the ATP that's available immediately available and present within the sarcoplasm. When it's consumed and can't be replaced by the generation of new ATP, then you turn to stores. And the way that the skeletal muscle stores ATP, or phosphate, high-energy phosphate, is in phosphocreatine. So when we are resting, there's a muscle called creatine kinase, or creatine phosphokinase, and the textbook describes how there's different isoforms of this molecule from skeletal muscle versus cardiac muscle. In this, there's a blood test to measure the amount of creatine phosphokinase, or CPK, or maybe called CK for just creatine kinase. There's a cardiac and a skeletal isoform. And so if it's up, then they want to find out which isoform is up. And if your cardiac one is up, you probably had a heart attack, right? If your skeletal creatine kinase, or CPK, is elevated, it means that you've got skeletal muscle damage or degeneration. Anyway, just a side note on the clinical relevance of this: it should only be in the muscle cells. When it's found in the blood, it's because of the breakdown of muscle cells. There's always a little bit there, but above normal levels is a clinical indicator of a problem. Okay, so back to what we are considering here after that clinical side note. So that the muscles at rest can generate ATP from metabolism from the glucose, and creatine is present. As you know, many sports enthusiasts, athletes, bodybuilders, people in general that want to be sure their muscles have got the energy that they need will take creatine as a supplement, thinking that when it's orally consumed, it can find its way into skeletal muscle and maintain the normal highest level, physiological level possible in the skeletal muscle. And then during rest, that will be combined with the creatine kinase and ATP to produce ADP and phosphocreatine. So as any kinase would do, when we studied the signaling, like the cyclic AMP activates the protein kinase A, then the kinase will phosphorylate proteins. In this case, it's phosphorylating creatine kinase, and ADP is the other product, right? And then this ADP is available to generate ATP, as you might know, or simply to reside in the muscle. It cannot be used by any other source, metabolic process, as ATP, but it can stay available for when the muscle is working, so that the phosphocreatine can be acted on with creatine kinase again to reverse the reaction, so that you get creatine and ATP. Then that ATP that would be available for the myotin ATPase contraction, as we had talked about when we looked at it in the previous recording on this, and the calcium ATPase, responsible for removing the calcium so that you get relaxation, and the potassium ATPase, as I had mentioned, in the membrane, to restore the ions across the cell membrane during the action potential to their original compartments. So contributing to resting membrane potential and regenerating resting membrane potential, such that you can have the membrane ready for another action potential. So what you're noting is that this is the same enzyme, and what it depends upon is the concentration of ATP. So if ATP is higher during rest than and creatine is available, it will make all the phosphocreatine that can possibly be stored as the ATP is produced. Now when ATP is used by other mechanisms, then it's going to decrease the substrate on this side, and then these will become the more active substrates to reverse this reaction and produce the ATP. So this is a freely reversible reaction that depends upon the concentration of ATP. As it's consumed, it's going to cause the reverse reaction. As it's produced under rest, it'll drive the forward reaction. So if that's not making much sense now, you should go back to the introductory chapters and read about the reversible enzymatic reactions, the general properties, and definitely by the time we start talking about muscle metabolism in our integration 2, not the upcoming one, but a later one, we'll revisit these properties of enzymatic processes. Well, quite obviously, it's possible to overcome both the immediate ATP resource and the creatine phosphate, the phosphocreatine source, and then the muscle begin to go into fatigue. So the calcium might be available, but if the ATP is going down, you're not going to be able to regenerate the resting membrane potential so well, and you're not going to be able to sustain myosin ATPase or remove the calcium, so the muscle's just not, it's going to get sluggish, right? That's the fatigue. The possible causes of muscle fatigue are shown in this slide. So there's different types, a central and a peripheral, and they're just basically what they mean. There's a psychological effects or protective reflexes within the nervous system will, of course, be impinging our abilities to trigger the somatic motor neuron to drive the system at the neuromuscular junction. There can even be neurotransmitter depletions at this level in extreme cases. And then the peripheral fatigue includes that exhaustion of the vesicular portion, to where the acetylcholine is being released more frequently than what the acetylcholinesterase and choline reuptake and choline acetyltransferase to replenish the acetylcholine can manage, or possibly even the vesicular recycling mechanisms and the recharging with the transmitters, what we talked about before. So the basal synthesis rate of acetylcholine, the recycling rate of acetylcholine, the recycling rate of the vesicles, and the recharging of the vesicles, the vesicular transporter to put the acetylcholine into the vesicle. All those molecular mechanisms can be overwhelmed in extreme cases. Either the duration of the physical demand or intensity, and of course both. If you think of a marathon runner or a decathlon events and things like that, over time these molecular systems can be overwhelmed. So not just the muscle fatigue, but the neural system can too. But the muscle will typically fatigue first, so there'll be a change for most of us that aren't extreme athletes. There'll be a change in the muscle membrane potential that can be fatigued because the ATP is not available for the ATPase. And there's other systems for membrane maintenance as well, but we're focusing on the ones that are known best for the membrane potential. The calcium signal, the sarcoplasmic reticulum has a calcium leak. Then there's the calcium release that would be decreased. And then the calcium troponin interaction will be decreased. And you get the depletion theories coming into play here, the phosphocreatine, the ATP, and the glycogen. And in the accumulation theories related to the change in pH with the hydrogen ion concentration increasing, making it a lower pH and acidic, right? The inorganic phosphate accumulation and certainly lactate accumulation. So these are obviously discussed in greater detail in the textbook, and I'll refer you there for that. We want to just make the point that there's a central and a peripheral, and there are molecular events that are impacted as we had discussed. So these causes, then we consider them during extended submaximal exercise with the depletion of the glycogen stores. The short duration maximal exertion will be related to increased levels of inorganic phosphate, and it may slow the PI release from the myosin. Remember, all these events of binding are concentration dependent. So if there's so much PI floating around, it's going to favor staying on the myosin head, even if the affinity decreases for the binding to it. If you have such a high concentration, it's going to keep bumping into it. So more frequently, it's going to be in the bound state, even if it doesn't want it there, right? It's like somebody spraying a water hose at you. Even though you don't want it, putting your hands up in front of you, it's still coming at you and sticking to you. Not the greatest analogy, but it helps. The decreased calcium release is related to the membrane property changes, as we had said. Then during maximal exercises, the potassium leaves the muscle fiber, leading to the increased concentration that is believed to decrease the calcium concentration. So the skeletal muscle during fatiguing submaximal exercise is considered in this slide. So on the x-axis, we see the muscle metabolites in the resting muscle and after cycling exercise to fatigue. So we're seeing the ATP level, the lactate level, the phosphocreatine level, and the creatine level. And on the y-axis, we have nanomoles per kilogram of dry weight of skeletal muscle. So they're taking a muscle sample and then drying it and getting a weight, and then they measure these compounds within it. They're not like cutting off the cyclist's leg. They're just getting a little piece of the skeletal muscle. It's possible to take a little sample. So the ATP they're showing did not appear to have any significant decrease. Certainly the lactate increased, as you would predict. The phosphocreatine after fatigue had a dramatic drop. We don't have error bars or statistics, so all we can say is it's a dramatic or obvious drop. The magnitude of the drop is from approximately 90 millimoles per kilogram dry weight to, per kilogram, they must have calculated up from a small amount, to about 65. So that's a robust drop. And then the creatine, paralleling this, of course, increased. So we would predict that the ADP concentration, that the ATP concentration being sustained at fatigue might be related to this phenomenon here. So the only other substrate reactant not shown here is the ADP, but we can presume that that would be following a similar pattern. So remember, the creatine plus the ATP produces the phosphocreatine. The phosphocreatine at times where there's an ATP demand, the turnover ATP is high, then this is going to be drawn to the creatinine and the ATP instead. So even though they're showing the static levels this way, the turnover of ATP is going to be higher, even though we have normal levels here during fatigue. So the fast twitch glycolytic and the slow twitch oxidative muscle fibers are shown histologically on this slide. So the slow twitch oxidative, there should be an O there, muscle fibers, you note the smaller diameter and the darker color due to the myoglobin in them. Those are more fatigue resistant. So the myoglobin helps them to store oxygen as well for these oxidative processes. And then the fast twitch glycolytic muscle fibers are larger diameter and a pale color, and they are easily fatigued. So there are other molecular and physiological properties of these muscles, but these are the points we just want to make on this slide of the diameter and the rate of fatigue. So smaller diameter with a slower rate of fatigue, larger diameter with a faster rate of fatigue, and of course the rate of the twitch. So this table summarizes more of the differences between the muscles and adds a third column. So there's the slow twitch oxidative red muscle, the fast twitch oxidative or glycolytic red muscle. Then there's the fast twitch glycolytic white muscle. So somewhat of a transition from slowest to fastest, and now having an intermediate for the speed of the development of the maximum tension. The myosin ATPase activity is slow in the slow twitch, fast in the fast twitch, and fast in the fast twitch. So the diameter shows the gradation as well. So the intermediate is the medium diameter. Contraction duration is longest in the slow twitch, obviously, and the too fast have the shortest. The calcium ATPase activity in the sarcoplasmic reticulum is moderate and high. So we have not yet found the major distinction for the intermediate one to differentiate it from the other two up to this point in our list. Endurance, we have fatigue resistant, and easily fatigued. So obviously the red muscle, the myoglobin, is going to contribute to the intermediate muscle being more like the slow twitch, as we see for fatigue resistance. For the use, the slow twitch oxidative is most used, and it is very important for posture. Standing and walking would be the intermediate, and then the least used, the jumping, quick, and the fine movements, would be the fast twitch glycolytic white muscle. Under metabolism, we have oxidative and aerobic, and then the glycolytic, but sometimes more oxidative with endurance training under the intermediate, the fast twitch oxidative glycolytic red muscle. The glycolytic more anaerobic than fast twitch oxidative glycolytic type of course contributes to the higher rate of fatigue in the fast twitch glycolytic white muscle. Capillary density is going to be high, medium, and low. So here, with a higher anaerobic level and a low capillary density, you get the typical buildup of lactic acid that still, I think, is a standard in muscle physiology for muscle cramping when you use muscles that are fast twitch to do these types of things that you haven't used for a while. So these can be conditioned as well, and vascularity can change, but the capillary density, relatively speaking, will always be lowest in the fast twitch muscles. The mitochondria are numerous in the slow twitch, and then moderate, and then few. So again, these all relate to the rapid fatigue. And then the color, of course, you have a dark red with the myoglobin, the red and then a pale coloration. And the final concept in this presentation before moving on to others is to pull together what was in the previous section. I just left these in the same sequence, original sequence. It seems to be a bit out of place, but it's a transition back again to talking about muscle twitch versus just muscle fiber twitch. And we can kind of bring the two ideas together of a single skeletal muscle cell and a whole skeletal muscle by saying that if we think about an individual sarcomere and the original stretch on a skeletal muscle cell or even a skeletal muscle, then we can imagine how if we stretch it too far before we try to generate some tension, there's just not going to be enough overlap of the thin and the thick filaments to do anything. And if we push it too close together, so we've got too much overlap, the maximum overlap possible, I don't care how much ATP and calcium you've got around, you can't get any more decrease in length. So we're going to have two ends of a curve where we would not be able to generate any tension, and then there's going to be every possibility in between. So whether you're talking about just studying a single sarcomere or a whole muscle. So if you put so much weight on a muscle that it stretches it so much, then you're not going to be able to generate any strength in that muscle to move the weight. So we can make that leap here. So quite obviously, knowing the typical relaxed state of a sarcomere and the maximally stretched and the maximally contracted, we saw those electron microscopes that can make the actual measurements, then it is possible to generate the curve of all the intermediate levels. So the optimal resting length is shown here at this position in C, and so our x-axis is the length of the sarcomere and the initial length, and then the y-axis is the tension and the percent of the maximum from 0 to 100. So the maximum tension that you can get is at the optimal resting length, where you've got a certain amount of prior existing overlap of the thick and the thin filaments, where if you've got the ATP and you've got the calcium, then you can get a contraction. If you stretch beyond that, from 2.3 micrometers out to 3.7 micrometers, there is roughly a linear decrease in the percent of maximum tension that you can generate. And then the slope, if you use the calculus to get the slope of this, or the rate at which you get this change, it is greater going from the maximally decreased length to the optimal length. I think you can see that a slope of a line here, if you forced a straight line there, would be greater than the slope of the line here. So there's something about the properties of increasing to maximum that differ from the decreasing from maximum. So you can change the length, although it's increasing it, a little bit more and still get a good tension out of it for a longer range, a wider range of increasing lengths, and still get some tension out of it versus your decreasing. So if you shorten it, you're going to get a more rapid decrease in the tension that you could possibly produce. So in other words, if you're 3/4 flexed in your bicep and you got a weight in it and you try to contract, you can't get the anywhere near the total 100% tension. You might get half the tension that you could if you started off with a nearly straightened limb with some weight on it, putting a stretch. So this is the leap that we want to make from a single sarcomere to thinking about the weight being put on an entire skeletal muscle.